Hello, and thank you for your interest in our current research paper. Allergen-specific immunotherapy is a causative treatment for allergic patients that induces long-term tolerance to allergens by reducing pro-allergic TH2 cells. However, only successfully so after at least 3 years of repeated allergen administration. This repeated treatment led us to ask the question whether the repeated antigen encounter drove the pro-allergic TH2 cells to exhaustion. The exhaustion phenomenon was first described in chronic LCMV infection in mice in the late 90s. When mice are chronically infected, their CD8 T cells become functionally exhausted and express exhaustion markers like PD-1 and CTLA-4. To address this subject, we used 3 angles. First, in a mouse model of allergic airway inflammation with allergen-specific immunotherapy, we found that both exhaustion markers were significantly upregulated on TH2 cells upon allergen exposure. PD-1 and CTLA-4 markers decreased after therapy, in particular on the surface of lung TH2 cells. Second, similarly, in a cross-sectional patient cohort, the expression of CTLA-4 and PD-1 and TH2 cells from patients with allergic rhinitis was enhanced compared to healthy individuals, with an even stronger effect in those with concomitant asthma. Third, in order to track these cells during the course of therapy, we analyzed peripheral T cells in a longitudinal patient cohort and discovered a late differentiated, pro-allergic, already exhausted T cell population with TH2A cell characteristics before therapy, which was initially reduced in the first updosing phase, but recovered and persisted long-term during the course of therapy. So what do we learn from this? Since these cells also retained their potential for self-renewal, it strikes us that their persistence may explain the long 3-year duration of therapy that is required to effectively reduce symptom burden. I would like to thank you for your attention.